Hello, good afternoon or good morning, depending where you are connected from. First, let me to introduce myself. This is Mabel Loza. I am professor of pharmacology at the University of Santiago de Compostela and lead of the screening platform in the Themus Research Center here at the university, which is one of the high capability screening platforms of the European ERIC e-Open Screen. I am also scientific director of Hertor Foundation, a foundation here in Spain, in Galicia, devoted to apply innovation to drug discovery. It's a pleasure to moderate this roundtable and to welcome all of you who are taking time out of your busy agendas to participate. As many of you already know, Keystone Symposia convenes some of the leading life science conferences worldwide. And this virtual Keystone Symposia ePanel event is part of an initiative to bring more science to more people, taking advantage of these difficult times for meetings in pandemic times. So it's focused in brain diseases, the diseases that have increased the most worldwide in recent years in terms of years lost and deaths. So despite significant advances in drug development tools, technologies, and strategies in recent years, these advances have not translated in real successful treatment for brain therapeutics. So we desperately need approaches to innovate the landscape of drug discovery in brain diseases, in brain therapeutics. And interestingly, the COVID-19 pandemic has yielded some critical perspectives, opening us a new window into how to think and to work differently to smash all timelines to drug discovery that, as you know, are typically in the classical all timeline about 10, 14 years for a new disruptive discovery, right? So we in this panel event continues the brilliant discussions initiate in a really pleasant roundtable during the Keystone Symposium on Brain Therapeutics, coordinated by Maria Blanco, Kalpana Merchant, and myself on February 2020 last year in Santa Fe, just at the beginning of the COVID-19 pandemic. So we have reconvened again our speakers, and we appreciate very much from them to do it. To continue this discussion, we started very successful in Santa Fe now in light of the new landscape shaped by COVID-19. And today we are really very happy to invite all of you to join to this brainstorming on an extremely urgent topic: how to innovate the landscape of drug discovery in brain therapeutics, taking into account lessons learned from vaccines developments in COVID-19. For this is this ePanel, and we will start with 40 minutes discussion among our speakers, panelists, followed by 40 minutes of live questions and answers with the global audience. So please submit your questions at any time to Q&A chat box, and also vote if you consider others' questions you would like to discuss. So now I would like to introduce our panel. We will proceed with a first quick turn of floor between all the speakers, and then we will repeat and start discussing. So we'll go to them.
Hi, I think Mabel, I think, am I first?
Yes, Sohini.
Just
So
want to confirm.
let me please introduce yourself. Sorry. As I mentioned, I will try to go from patients to therapy. So first is going to talk Sohini Chowdhury. She's a deputy CEO and head of research for the Michael J. Fox Foundation. She's a young, as you can see, brilliant innovator that changed the shape building of drug discovery with an unprecedented stakeholder collaboration she established for her institution, patient focused and patient solved and problem solver. So it's really a privilege to have you here today, Sohini. Thank you very much.
Thank you for inviting me again, and I'm really excited to continue the conversation in light of the 18 months that has passed since we all last met in Santa Fe. So thank you again to the organizers and Mabel for this panel. Sorry, Mabel, do you want me to make a few opening remarks, or do you want to go to the next individual?
No, I would like from you to have your view in, as we mentioned, about 5 minutes, and then we will pass for the others. Thank you very much.
Right. Sorry, I didn't catch that. Thank you. So, so as Mabel said, I think I present a unique view amongst this panel in that the Fox Foundation, for those who don't know, is an organization that was founded 20 years ago with one real mission in mind, which is to aggressively fund research to accelerate the development of impactful therapies for people with Parkinson's. And I think when we look back, it's 20-year anniversary. And when we look back as an organization, we sort of see the trajectory of drug development in Parkinson's. It's actually, it's pretty mind-blowing. When you think about it 20 years ago, the landscape for Parkinson's drug development was very much a landscape of Me Too drugs, focused primarily, if not exclusively, on addressing the symptoms of Parkinson's disease. And by that, a very limited set of symptoms, the motor aspects of the disease. Fast forward to, September 2021, and it's a very different drug development landscape. And it's very much predicated now on exploring different avenues, whether they are specific targets of interest, such as alpha-synuclein or LRRK2 or GBA, or pathways or mechanisms that are of interest and have been, highlighted as related to Parkinson's disease, so inflammation, etc. And I think that the question is, what happened in 20 years that changed it from a Me Too kind of very limited sort of focus to really, honestly, a golden era, if I could call it that, of Parkinson's research. And I might be a little bit controversial here, but I will say that I think one of the biggest things that changed it, and I think it's a little bit where I'm going with the perspective that I bring to this panel, is that it's a little bit about genetics, and not about genetics per se, but about going back to the patient and understanding what can we learn from the patient. And I think genetics is the tip of the iceberg. I think genetics is probably something that is the easiest for us to put our hands around. You can identify, mutations of interest, etc., and then it leads to the building blocks that sort of expand that knowledge. But at the end of the day, I think genetics was a bit of a catalyst. It allowed us to begin to say, yes, synuclein or LRRK2 or GBA, these are things that we know are definitively linked to the disease that an individual is experiencing, the pathological processes that are part of that disease. So let's start to work in that. And it begins to blossom from there. And I think one of the roles that an organization like the Fox Foundation and others that work in different disease areas that do similar things that we do within Parkinson's is to kind of bring it back to the patient. How do we continue to support drug development efforts by providing the necessary samples of individuals with the disease across the disease spectrum, from those that have had the disease for multiple years to those who were just diagnosed to those who actually may not even present with symptoms yet, but may be classified as risk for developing the disease. How do you get those samples? How do you get the corresponding clinical information, et cetera? Because I think at the end of the day, you can have the greatest technology, and admittedly, advances in technology, again, around sort of genetic testing and sort of genotyping, et cetera, have been critical in the advancement of drug development. But if you don't go back to the patient and you don't have the source, the direct source, of information about the disease itself, I think you're always going to be limited by how much progress you can make. And I think I'll end it there, but I would say that, at least in my mind, Mabel, you laid it out so beautifully in your introduction. If you look back at COVID, I think one of the beautiful things about how fast we saw drug development go with COVID is that we were able to get information from patients. We were able to kind of have doctors share what they're seeing, the symptomology of the patients, what they were experiencing, how they were reacting to the different, especially in the early days, the different approaches towards treatment, et cetera. And those became the building blocks. And I think that's one of the things I would stress in this panel, is not to forget that we have to keep the patient in the center of our research paradigm.
Thank you very much, Sohini. Thank you so much. We will come back to these words for discussing. So we will continue talking about how to connect faster, to create innovative connections between basic and clinical science, both in models, technologies, but ways to do it. So we have here James Audia. He is owner and president of Audia Consulting, a distinguished scientist and also founding scientist at Therapeutics, and he's also senior advisor of Karuna Therapeutics, a visiting scholar at Northwestern University and adjunct professor at the University of Illinois Chicago. He has a long and distinguished career in the pharmaceutical industry as prolific inventor with more than 110 issues US patents, and he also made with different companies the road from the basic science to advanced clinical developments, the one that we are thinking now to do faster. So we are very proud to have today his broad and brilliant expertise team. Thank you very much.
Thank you very much. Thank you very much, Mabel. And I'll reiterate what Sohini said, that it was such a pleasure to get together 18 months ago at the Keystone meeting and try and recapture some of the spirit of that discussion, the camaraderie. I think that's one of the things that we've really missed during these 18 months, during the pandemic time. But it also gives us a snapshot in time, pre-pandemic to today, and a number of remarkable things have happened. As we think about brain therapeutics, and perhaps this is controversial, but there's now been a drug approved for the treatment of Alzheimer's disease, right? And so this is remarkable in and of itself, and it's, I'll withhold comment about, my own thoughts about that approval, but the consequences are significant, right? And there's clearly been a renewed vigor, a renewed appetite for investment in neurodegeneration research, neuroscience research. There have been a number of new companies formed focused on neuroscience and neurodegeneration. There have been, and I think over the course of this last 18 months, by virtue of having to operate in a virtual environment, I think there's been a democratization of science. We're no longer restricted to well-defined geographic boundaries and hotspots for doing research, but we're able to collaborate broadly. I think we've learned how to work with colleagues at great distances, and that's become normal, right? I think we've really normalized the ability to interact with colleagues. I'm involved with their Flare Therapeutics, a new company that has embraced a virtual, a hybrid virtual in-person working model. And we're recruiting people from all around the world, realizing, that we've learned how to collaborate effectively and interact effectively without being co-located. And so I think it has opened up a number of possibilities. I think the other thing that's been remarkable is just the continued pace of progress around new tools that, again, I think many of these tools existed in the past, but they were always limited in their application. They were either not applicable to neuroscience problems, or they weren't available to everyone. They were only available for expert users. And now I think there have been exciting breakthroughs. The DeepMind AlphaFold, which has been able to predict structures of really all of the proteins that are encoded within the genome, and has made those structures available in an open source fashion. Virtual screening platforms, again, available open source, that have democratized the ability to screen for chemical matter that engages or has the potential to engage targets. Structural methods that for many years were really not applicable to many neuroscience targets have now produced really interesting results. And so many neuroscience programs, in fact, are structurally enabled and structurally guided. Machine learning and artificial intelligence have been, I think, successfully applied to so many different types of data, whether it's genetic data, it's patient data, applying it to behavioral data to try and glean out patterns or signatures that help us guide drug discovery efforts. They've been involved in or been applied to the design of new molecules or the interpretation and coalescence of data. And so I think it's been an incredibly exciting time to be doing neuroscience research.
Okay, thank you very much. Very good point. Next, we are passing to the next speaker. In the current pandemic, we have realized that the critical role of the scientific journals to facilitate this breakdown of many barriers that Jean mentioned, to facilitate this global communication of newest scientific and technical advances. For this reason, we introduced today a new speaker in our roundtable of Santa Fe, which is Julia Meliak. She is scientific editor at Disease Model and Mechanism, a company of biologist journal. And also very important for us, she made a brilliant scientific career before to move to the journal. And as I mentioned, very important for us too to have she in our symposia, because she very actively participate in discussions in, as I mentioned, Santa Fe Keystone Symposium. So this time we invite her to join us about the publisher's critical expanded role during COVID to facilitate this advanced by promoting the open science. I'm very well connected with the talk to Jim. Thank you both. Yes, Julia, Thank you very much for being here.
Thank you very much for the invitation, Mabel. It's been a pleasure staying in touch since Santa Fe and obviously thinking about a follow-up discussion. So, of course, the scientific publishing landscape is changing. One of the things we have to think about is that it's not enough for journals just to be places where someone puts their paper. We need to be hubs where communication is encouraged, where people from different specialities talk to each other. So Hini talked about the importance of going back to the patient, and she talked about how important it is to understand the mechanisms, the function of the genes behind a phenotype that we see in patients. So I think that this is where open access scientific publishing can be that key missing link, because of course, in order to understand how variants of genes that are implicated in disease work, we still need to do that in model systems. And traditionally, people who played with model systems in a lab were very separate from people who were sequencing genes or from people who were actually treating patients. Now the world has changed, following up again on things that Jim said, and Suhini said technology has advanced, and clinicians have to talk to people in the lab and have to talk to bioinformaticians all the time. Thank goodness, although we can't be together in the same room, we do have tools available to us. There are specific tools on the internet, such as Model Matcher, et cetera, that allow clinicians to actually find whether a lab somewhere in the world has developed a specific model system tailored for that particular mutation that they identified in a patient, which is amazing. And then when it comes to, of course, sophisticated model systems that we can use for personalized therapies, where we can actually recapitulate 4 or even 5 different mutations seen in a patient in a very easy to manipulate model platform, in which we can then screen hundreds, if not thousands, of compounds to see which one will work. We are understanding very, very deeply how genetics affect the phenotype in a patient, from a molecular level to a cellular level to the organism level. Again, we are understanding that when it comes to brain therapeutics and the brain itself, it's not just about the neurons and the non-neuronal cells. It's about processes such as inflammation, metabolic alterations, and all of that. So I think here is where journals can actually be active and participate in conference, help scientists organize conferences, help them talk to each other and break the silos so that funders and patients and scientists and clinicians can actually have a common language, and this language can live and be created with the help of journals.
Thank you very much, Julita. It's almost just a discussion working. So, and now to finish this first round of words, the capability to act on time to answer to the urgent needs. For instance, the Oxford AstraZeneca vaccine, right? So Charles Bontra, who of course was in the first roundtable in Santa Fe, is Pro-Vice Chancellor for Innovation at the University of Oxford, Professor of Translational Medicine and Director of the Center for Medicines Discovery. Previously, he was Vice President and Head of Biology and GSK. He has awards and country and public acknowledgement and countless, and he is recognized as top innovator, leader, and inspiration for all of us. And also, again, protagonist, as I mentioned, in some new vaccines discovery. So we expect a lot from you, Chas. Thanks so much, as always.
Well, Mabel and colleagues, thank you. Mabel, you're excessively generous. That's very kind of you. But thank you for allowing us to take part in this sort of follow-up discussion. What I'll do is maybe just share with you a bit about my background and maybe some of the lessons, at least I've learned in the past 18 months or so, and I hope they might be useful for our discussion later. So, I had the pleasure of working inside GlaxoSmithKline, and for many years worked with some very smart people to try and develop drugs for neurological and gastrointestinal diseases. And of course, we identified many targets. We generated high quality clinical molecules. We took many of these molecules into the clinic. Many of these molecules demonstrated superb data in animal models, etc. But invariably, when we take things into the clinic, things don't work as well as we would expect or we would hope. And of course, we ask ourselves questions around, have we got the right target, the right dose, the right biomarker? Have we stratified the patients appropriately, et cetera, et cetera? So I left GSK in 2008. I came back to Oxford, and for many years, what we did was we, as Sahini was saying, we identified novel genes that had come out of large-scale human genetic studies and which had been underexplored or not explored at all. And then for those genes, what we did was we generated very high-quality tools. So, for example, we purified the protein, or we built assays, or we built structures, or we generated small molecule inhibitors and antibodies, etc. And these were high quality because we were working with several pharmaceutical companies and several patient groups. And then, but all of these tools, what we did was we made them freely available. So we shared them with the world. We worked with lots of academics all over the world to take these tools and explore new biology and hopefully identify or validate new targets for drug discovery. Now, that open science has been phenomenally successful. You know, this was a way of bringing together lots of academics, lots of companies, lots of patient groups, and trying to work together to reduce duplication and identify truly novel targets that industry could take forward. About 6 years ago, what we also did in Oxford was, again, working with a patient charity, this time Alzheimer's Research UK, we started a program here to build a pipeline of new targets specifically for dementia. And we recruited a brilliant guy who I used to work with at GSK named John Davis. And John has worked with many colleagues over the past 6 years. He's leveraged lots of academic resources, lots of clinical resources. He's built great partnerships with large pharma companies and biotechs. And now 6 years on, we have quite an exciting pipeline of new targets for dementia. As Mabel said, we're now building in Oxford a new institute called the Centre for Medicine's Discovery. And our intention is now not just to generate novel tools for genetically identified targets. We now want to work with industry, venture capitalists, philanthropists to generate clinical molecules and then work with local academic clinicians to take those molecules into patients, into phase 1, into phase 2 studies. Because, of course, ultimately, that's where we really find out if molecules are effective. So that's a little bit about the sort of background. But let me just share with you some of the lessons from last year. Now, again, you know, I've had the privilege of sitting here in this amazing university. And if we cast our mind back to, I don't know, March, April last year, you know, many of us were locked at home. The elderly and the vulnerable were scared to go outside. We had no treatment. We had no vaccine. Many people thought that a vaccine wasn't even possible. But let's just look at what's happened in the past 18 months or so. So my senior colleagues here, I mean, Louise Richardson, who's our Vice Chancellor, John Bell, who's our Regis Professor, and an absolutely amazing guy, Gavin Screeton, the head of our medical school, worked with Adrian Hill and Sarah Gilbert, who had created this company called Vaccitech. But they pulled in colleagues from AstraZeneca. So Pascal Swaro, the CEO, and Mene Pangloss, the head of R&D. They pulled in people from the UK regulators, from the UK government, and from several funders. And they sat down and said, how can we generate a vaccine as quickly as possible to save the planet? We're not interested in making money. We want to literally save people's lives. And it is quite stunning to me what my colleagues did in the space of 9 months. You know, in the space of 9 months, they created a vaccine, demonstrated efficacy in the clinic, got approval, etc. And since then, since approval last December, that vaccine has been, I think now, into close to 1.5 billion arms. And that vaccine is also being distributed at cost price. So my applause to Pascal Swaro for having the courage to do that. AstraZeneca in the past 18 months have made no profit from giving 1.5 billion samples of this vaccine into patients. I think it's absolutely laudable, and I would suggest that Pascal deserves some sort of recognition and probably the Nobel Peace Prize. I think it's a phenomenal thing that he's done. But for me, the big lesson from all of this is that when you get all the stakeholders together in one room and you create that single vision, that single focus, that single goal, we're going to generate a vaccine for this virus, COVID-19, as quickly as possible. It is amazing what we can do. We really can do the impossible. You know, collaboration is key, but we can do the impossible. The other thing I would just share is that money was not limiting. You know, we fortunately had lots of funding from AstraZeneca and the UK government and many other funders, etc. Money was not limiting. The third thing I would say was the goal last March or April, it wasn't, oh, let's generate a vaccine for every single virus on the planet. It was specifically COVID-19. So I think focus is incredibly important. So here we're talking about brain diseases, but you know that those are so many different diseases. We know each of them are so difficult. We're not going to come up with a therapeutic for all of them in the next 9 months, etc. So if we really want to do something and we really want to accelerate, we're going to have to come together, focus, and ensure that funding is not limiting. The other couple of brief stories, again, you know, a couple of my colleagues here, Martin Landre, Peter Horby, brilliant clinician scientists. What they've done is they took existing drugs and looked to see if they would be effective in COVID infections. And of course, they tested these molecules in tens of thousands of patients, accessing lots of hospitals and patients in the National Health Service here in the UK. And they demonstrated unequivocally that dexamethasone, this cheap generic anti-inflammatory that's inside every pharmacy shop, clearly works in COVID infections. It has saved more than 1 million lives already, more than 1 million lives. Now, they also demonstrated that hydroxychloroquine, which the former resident of the White House was telling us would be effective, that had absolutely no efficacy. So again, it's great how sort of great clinician scientists working in academia, accessing hospital, clinical, patient resources, can do things at scale and at speed. And the third thing I would just share with you is some of my colleagues here, Dave Stewart, Yvonne Jones, Frank Vondel, etc. They did this program called Moonshot. And so what they did was they wanted to generate small molecule antiviral drugs, again for COVID-19. And what they looked at the main protease. They screened this protease using crystallography. They identified a number of chemical starting points. And those chemical starting points, they shared them with the whole world because they recognized that they didn't have enough chemistry resource themselves here in Oxford to take those forward. They shared them with the whole world. And then chemists in South Africa, in Brazil, in Australia, in India, all over the world, use their own resources to take those starting points, to optimize them, and very quickly they generated very potent nanomolar inhibitors, etc. So this is just another example of what, if we do things in the open, how we can almost crowdsource science. And that's what we have to do. If we have an urgent problem, we need to come together, we need to work at speed, and funding should not be limiting, but we just have that single goal. So, Mabel, I'll shut up there. So thank you for your time, and it's a pleasure to see you all.
Yes, thank you very much, Chas. Really, we expected, but we have it. So it's very interesting to listen to you. It's clear that you, as very smart and very centered in the world of drug discovery. People knows all the details, but one thing that we didn't not mention only, Jim, is that there were also an inconvenience that was the focus in COVID reduced the dedication, both sometimes in thinking, but also in investment in brain therapeutics. So this is the negative part. It happens with another common diseases with the same. But I would say that you all agree with different perspectives that the focus is the world that help to have this incredible success in COVID vaccines. And the focus in patients, in the details, all is always in the details, in the targets, in the use of the info in this democratized scientific world, like Jim says, and is open, like Julija. The info of the patients, of the therapeutic results, of the evolution, of the new possible drugs, scaffolds, and take advantage, as Chas says, of them to have a better therapies and to know how to use the current therapies for this. But my question is to come back to second question we discussed in Santa Fe, and to the light of all this new world that is clearly different. I would like to listen from your point of view, focusing as just say, how to scout this, we can say, global democratic ecosystem for transformational innovation, for efficient way to do that, to deliver new targets linked to disease, to the biology, to the data, the new big data that we have, the associate markers to select the right patient, as Sohini said. How do you think that we can improve this not really impressive success in central nervous system therapies with all these things? Do you think that it would be possible to differently start looking for this? Do you would be able now to start defining? We can see, okay, these diseases are also urgent needs, right? For people. So how do you start a similar global concentration to deliver something faster, right? Each one of you, as you prefer. If not, we can start another round of words.
I mean, I think it's, I think focus is absolutely essential, Mabel. I think, how does one, I think with COVID-19, it was easy to find the motivation for that kind of focus and a global effort, right? This was something that, that had the potential to impact every person on the planet. And so I think it became a somewhat simplified task to really mobilize all of these resources on that. And Chaz is correct. We talk about brain diseases, and there are so many, and they all, how does one choose, right? On which disease to focus? How can you mobilize a COVID-19 like effort on any one disease? I think that's clearly a problem that goes beyond my ability to comprehend. I do feel like groups like Michael J. Fox, the Rainwater Charitable Foundation, the Alzheimer's Association, I think they bring some element of focus. It's not necessarily at the global scale that existed for COVID-19, but I think they have been lenses, if you will, that can bring together, the patients, the caregivers, the clinical scientists, the basic scientists, along with the others in the community that can really provide a local focus that simulates what went on a global level for COVID-19.
Thank you very much, Jim. I would say that listen to you. I would ask Chas now about this because we are talking about focus in a world of a lot of information. We say that too much information sometimes generates loss of information. How to use all this information? So did everybody have hands? So I would go. How to generate small, I don't know, small factories of validation, looking at patient of new targets. Chas, and then Sohini and Julia. We have 10 minutes more.
Thank you, Mabel. I think leadership is critical. I think many of these individuals I mentioned, they are great leaders, they are great innovators, and they are great entrepreneurs. Once they get something in between their teeth, they're never going to let them go. And, that determination, that urgency, that risk-taking, you put a hurdle in front of entrepreneurs, they just get around it. That's what we need, those sorts of individuals. Secondly, I think let's be humble enough to realize that no great university, no company on the planet has got all the resources and the expertise that they need to do to crack any of these problems. It's absolutely clear. I think we need to start thinking much more international. I talked about university working with regulators, industry, government, funders, etc. But these problems are global. They're in the US, they're in the UK, they're in Spain, they're everywhere. These are global issues, etc. So we need to come together. But please bear in mind, each of these problems requires a ton of money. And, no one government is going to put enough into it. We're going to have to work together and we're going to have to focus. But that's great leaders can provide that focus.
Thank you very much. Good point. Sohini, please.
Yeah, I was going to say that I think the COVID example is really interesting, but I think we have to also recognize it's not an example that we can duplicate. It's very unique. The amount of resources and prioritization that supported the efforts across governments, across institutions, I think it's naive to think that can be replicated. I just do. That being said, I do think there's a lot that we can learn from that example about what is scalable and what is applicable. And in my mind, there's two items that I think are really important. The first is the sharing of information. If I remember correctly, and I may have the date wrong, but I think, we started to hear about sort of COVID in China and Wuhan, et cetera, around in December. And I believe that the virus was genotyped in January and it was published and it was shared. Think about that. You already started to get the sharing of information and it just built upon that. And you started to have everyone sharing. Now, the good is that you were getting enormous data available to great minds across different fields that could all sort of focus on it. The negative is some of the data were questionable, but I think you have to take with the good with the bad. And I think in this case, the good trumps the bad. This concept of get the information out there. Maybe it's not great, but we're going to trust the community to help vet that, and that the good will rise to the top and we can continue to build on the findings and the positive information coming out of it. And I think it would be fantastic for the CNS field to take that approach, which has already been sort of established in physics, right? And in mathematics, people publish, they go out there and they share the information and people then build on it. And I think while, I think it's also kind of naive to think we're going to be focused on one disease, we're not. But CNS, at the end of the day, it's about the brain. And there is a lot you can learn that either discounts things and takes things off the tables because there are findings in other areas, or it helps you identify new pathways. So, we learn a lot in Parkinson's from other Parkinsonisms, like tau or whatever, tau pathways or PSP or whatever. We learn a lot from Alzheimer's. We learn a lot from ALS. We actually also learn a lot from autism because there's a lot of just basic biological knowledge about the brain that helps our understanding. But it's not so easy to access that. And so I think here it's about really as a field, as a CNS field, and as all the major players, whether it's publications, whether it's funders, whether it's other institutions, maybe universities, making the effort to say to researchers, publish it, make it open access, share your data so that everyone can see it. The second thing I'm going to say that I think we learned from COVID is that it's not one field that is going to make the breakthrough. And I think others on this panel have already highlighted that, but I think it's really important. In Parkinson's, you know, there's a lot of great biologists, basic scientists, but and there's a lot of great data scientists, and there's a lot of great data generators. But if they don't come together with the clinicians who understand the disease, it's not going to move forward. You have to actually bring people together across the field. And I think that's getting easier, but it still requires a proactive push and an incentive to cross those silos, those natural silos of the basic scientist to the clinical researcher to the data scientist. We've had a lot of, you know, there's a lot of data out there, and that's great, but to translate it into information actually requires a team effort. It doesn't require, you know, just Watson. Watson is not going to be able to put the context and the needed to understand the disease and the implications. And so I think the other thing about COVID is that it was a real multi-stakeholder effort, from clinicians to patients to basic biologists to geneticists to, you know, public health officials. It brought everyone together. And I think you need that. You need to figure out how to incentivize and then support groups to come together in a multi-stakeholder fashion to make progress. So I'll end it there because I know we're running out of time, and I know Julia had her hand up first.
So thank you very much.
Thank you so much, Suhini.
Well organized, Julia, go on, please.
Yeah, thank you. So I agree with Suhini that COVID was such an unprecedented thing that had such a huge impact on every single person on the planet. And of course, there are lessons to be learned. But one of the things that we also saw in the last couple of years is the power of patients and their caregivers in the field of rare diseases. You know, if we look at spinal muscular atrophy, it's a very rare disease, but it was the patients and their mothers who pushed and learned to read papers and to understand the science, even though they were not scientists themselves. And they went from door to door, from geneticists to bioinformaticians to clinicians to then researchers and even companies. And now we have drugs for those kids. So I think that in the field of SNS, just like Suhini said, you know, it's not just one way of doing things. We can learn from big things like common diseases like COVID, but we can also learn from those one stories, N of one stories of patients and their families. Of course, this is very limited, and it does involve an element of privilege because those families have to have certain access to resources, but we can learn from those examples and we can bring people together the way those moms and those dads did.
So I think interesting and agreed discussion. So it's perfect time to go to open this discussion, this brainstorming to global people in this new way of discussing science. This is the good point also of this video conference. So we are starting this part of live questions and answers with 40 minutes more discussion. And for this part, let me to introduce the original coordinators of the Santa Fe Key Store Brain Therapeutics in the discussion that sparked this event. That are Kalpana and Maria. They and we will join forces again to coordinate this part of discussion. So I open the floor to them. First, Kalpana Merchant. She is adjunct professor for neurology at Northwestern University and president on Transterra Consulting provides strategic advice to companies in drug discovery. Also senior advisor of the main initiatives in the area. She also have an important experience in pharmaceutical companies, so have a privileged vision on the landscape, and it was and it is a privilege for me to share the coordination with her. So Kalpana, go on.
Thank you so much, Mabel. It's been a privilege to work with you for this Keystone Symposium that we had last year in Santa Fe, and then continuing with this panel. So thank you for partnering with me, and congratulations on a fantastic symposium and the panel today. I'll just take a couple of minutes of sharing my thoughts and then go on to Q&A with Maria. I feel like, as Jim brought up, that first time we have an approved therapy for Alzheimer's. But if you also look at simply the last 3 years of approvals within the USA, the FDA approval, unprecedented level of innovation can be seen in things like SMA that Julia brought up, Duchenne muscular dystrophy, migraine. If you see the treatment of migraine, we had nothing 5 years ago. And now we have all kinds of modalities, from small molecules to antibodies, a new drug for schizophrenia after how many years of using the same me toos that was brought up early on. So the modalities available for CNS, from small molecules to gene therapy to even cell therapy for ALS that's under development, as well as for Parkinson's disease, these could not have happened without the biological innovation, of course, patient-derived information, including genetics, but it goes beyond genetics in the sense of the biomarker work that has happened on patient-derived materials, be that cerebrospinal fluid or peripheral blood-derived cells. So I think it's an exciting time. And there are times when I discuss with my children, if I could turn the clock back, I would love to go back to graduate school and jump into this basic biological questions that we are able to answer with the technological innovations we've had just in the last decade or so. So with that, I think the panel today has addressed some very, very important questions. One being the focus, leadership, team effort. But maybe I should hold off, let Maria introduce herself, and then we can jump into the Q&As that have come through on the ePanel. And please continue to put your questions there, and we'll just go down the list. So Maria, over to you.
Okay, so Maria Jesus Blanco, Chus Blanco, here in Spain, in Galicia. She's a senior director and head of chemistry at Sage Therapeutics. She is a brilliant medicinal chemist and strategic leader, both directly in the industry where she was connected for many years, but also connecting with academias, connecting with journals, organizing editors. She is a really good example of these people, inclusive we are talking to, with more than 10 years experience in this, including pharmaceutical company, as I mentioned, also from Kalpana as director of discovery and chemistry research. She was the main organizer. She was the original idea of go on with this keystone on brain therapies in Santa Fe. So Maria, go on, please. So it's your floor, Kalpana and Maria. It will be connected, but it's in your hands, essentially.
Thank you, Mabel. Thank you for the nice introduction. It's a pleasure for me to be here, surrounded by such important leaders and continuing this important discussion, I think that around brain therapeutics. Some of my thoughts, we talk about focus. I think that, the COVID lessons was very important to me. Is not only focus, but a sense of urgency, right? There was an important disease that we need to together to come up with a solution because it was impacting everybody, as it was discussed. So my perspective is, are we having enough, sense of urgency about brain health? And I started seeing in the society a little bit more kind of in the open field, the importance of prioritizing brain health. I think that oncology, cancer, it gets many times in the news, but the importance of brain health, and many times the well-being starts, some of the diseases start 20 years before that you start seeing any symptoms. So how we can educate people, how we can be aware of this, and then come up with therapies. I love what Chad was saying about, bringing leaders, having tools, increase the sense of urgency across countries to put enough funding so we can make a difference for patients. So with that, I'm happy to coordinate with Kalpana about the Q&A. Please put your questions in the chat.
So there are several questions in the chat, and thank you very much for those who have asked the questions. I'll start with the first one that got a number of votes. This is from Jose Bria, who's commenting on the focus being a key driver for innovation. But this is a really good question. How can this focus be achieved in the variety of diseases we have that are brain diseases, when some of the diseases are based on what I would put as non-objective ways of diagnosing patients. So basically patient-reported symptoms, right? And so, and obviously this refers to psychiatric diseases. What can we do when there is such diffuse information to understand the disease, and how can we differentiate amongst these diseases when the symptoms many of them are commonly shared across the diseases? So perhaps Jim, if you want to start, and then others can add to that.
Yeah, I mean, it's a great question and a significant problem. To me, the path for success has really been to try and integrate data from a variety of different dimensions. I know that within, the Alzheimer's community, the ability to integrate imaging, biomarker, as well as genetic and clinical behavioral information, I think has been able to produce much more actionable diagnoses, much better stratification that I think starts to make sense. I think relying on any single data type, I think it always creates challenges, right? I think we have a desire to make things reductionist, but as someone smarter than me once said, make things as simple as possible, but not simpler. And so how do we add in the necessary complexity in order to really make meaningful diagnoses that are then actionable from a therapeutic, a treatment perspective?
Just to add to that a bit, the learnings that are coming from fields like Alzheimer's, where we have a number of biomarkers that are now used for drug approval. If you follow the literature in the psychiatry, same kind of technologies are being applied. For example, the blood-based extracellular vesicles carrying certain mRNAs or microRNAs or neuroinflammatory markers that a lot more work has been done in neurodegenerative diseases. People are seeing the success of that and are applying it to it. So I think in our lifetime, in the next 5 years, we'll see that groundbreaking research coming also into these so-called more diffuse diseases. Maria.
Okay, so I'm going to continue with the questions. The next comes from Francisco Javier Fernández Gadea. And the question is, in your opinion, which is the main roadblock today for fast and efficient open innovation initiatives in the CNS field? Perhaps Chas, you want to take that one?
I'm happy to have a go, Maria. I mean,
Which is the main roadblock today, nowadays, for a fast and efficient innovation? in the CNS?
I think we all know that the benefits of open science, and we also know the limitations of it. But I think the onus is on us to come together to create these ideas. The challenge we've got, of course, and we've all touched on it, I mean, there's so many diseases, there's so many technologies, there's so many modalities, there's so many molecular targets, there's people working on devices, diagnostics, different animal models, etc., etc. And the problem is the limited funding that we have for CNS disease, and it is less than oncology. And I do believe drug discovery for brain diseases is more difficult than drug discovery in oncology, etc. That small amount of funding we have just gets spread so thinly. So I think the onus is on us. I don't think there's a roadblock. I think the onus is on us to come up with ideas that we're going to work together. We're going to focus on this. We're going to make all of our data available, and this is going to catalyze the discovery of new medicines in Alzheimer's. I mean, obviously I was pushing the focus argument, but let me just share this. I mean, how much money have we spent in academia, in biotech, in industry since the mid-90s on the anti-amyloid approach? And, we've been going for nearly 30 years. Biogen get their drug approved, and there's still a lot of people who are not convinced about its efficacy. So, there you could argue we plowed tens of billions into that pathway, into that target, and there's still people questioning it. It just goes to show how difficult it is to come up with drugs for some of these indications. I mean, treating Alzheimer's is not going to be easy. We all know that. You're absolutely, somebody said, at least with COVID-19, you get an infection and a few days later you can see the symptoms, and, people end up in hospital or seriously ill, and unfortunately some of them die. With a lot of the diseases we're talking about, many of them are developing over years and decades, probably since the time you were born. And you don't often see the symptoms, or, they're not easily visible. And so we have issues around biomarkers and stuff like these. These are very tough problems. But I think the onus is on us to come up with ideas to create these open science networks.
Collaboration networks. I see they have their hands up, Julia and Jim. Let's go first with Julia. Go ahead.
Yeah, my comment would be that, of course, making drugs and finding markers by which we're going to even see whether our drug's working or not is important. But when it comes to the CNS, one of the things that is, in my very naive opinion, one of the biggest challenges is, once you have a drug, how do you get it into the brain safely so that it does not affect other organ systems, and that it is not in itself an invasive and potentially dangerous procedure. This is something that clinically is probably one of the things that cancer and infectious diseases don't have to deal with at the same scale as brain therapeutics.
Right, Jim?
Yeah, I guess the point that I was going to make is I don't know that there is a singular roadblock. I think it's actually a conspiracy of a number of roadblocks that exist. I think, I agree with Chaz that, many neurodegenerative conditions, many of these diseases are complicated. I think we have imperfect models of the disease. We rely on animal behavior to try and predict, much more complex human behavioral abnormalities. So, I don't know. I often think part of the challenge is that there's not just a single problem to solve. There are a variety of problems to solve. But I'm encouraged. There's renewed investment in neuroscience, which I think takes on one of the problems. I think there's increased molecular thinking. There's, biomarkers, genetics are being much more translated from oncology and other therapeutic areas into the study of brain diseases. I think we're able to both evolve better animal models, but also learn the limitations of the over-reliance on animal models and do more translational research clinically, which I think has been one of the real catalysts for the progress against COVID-19. Chaz talked about some of the studies, where, the dexamethasone data, where you're able to run very quickly well-constructed, well-considered, and powerful clinical studies to answer specific questions. And I think we're doing more of that in neuroscience as well. So I don't think it's a single problem, but I do think we're making progress on the many problems that have gotten in the way of neuroscience therapeutics.
Thank you, Jim. I do think it'd be important also to pair this, the kind of discussion we are having with the changed kind of approaches and systems through which we are developing drugs. So maybe both Jim and Chaz can respond to this on the role of the smaller biotechs in taking risks, in coming up with novel ideas on how to go after CNS diseases, and then how the development process then takes place through the large pharma that is usually not so interested in taking the risks, but they jump in at the right time point, right? Jim, you and I both have had the privilege to work with these small companies, but I do think that's changing how we are developing drugs today compared to how we were developing drugs even 10 years ago for CNS. So maybe you could comment, one of you can comment on that.
Yeah, you want to go ahead and then I'll follow.
Yeah, thanks, Jim. I mean, all I would say is sort of, in the past year, how much has the value of Moderna grown? Or how much has the value of BioNTech grown? And these are companies that focused on, mRNA vaccines or, what BioNTech were doing in Germany, etc. And literally, okay, opportunity presented, they focused, they scaled up, and they delivered. And so, even the Oxford example, the platform that Sarah Gilbert used to generate the vaccine was a platform that she had been working on for many years. So again, you could argue it's opportunistic. So I think what we've seen is, what I'm trying to say is, one success can convert a biotech company into a big pharma. Moderna is worth now, I don't know, is it 150, 160 billion? It's a big pharma company.
Mm-hmm.
Yeah, it's hard to disagree with that. My thinking about your question, Kalpana, though, I've seen movement, over the past 5 years about more startups in the neuroscience area
Exactly.
than occurred in the past. Sage was clearly a pioneer, started a decade ago, but there have been more neuroscience focused companies.
Exactly.
It used to be, within the VC community, lots of oncology, gene therapy, orphan disease kind of focus. But I think there's now a growing belief that companies focused primarily in neuroscience are capable of being successful. And, you've got different models. You've got companies that really only want to do the early discovery or potentially bring a molecule forward as a development candidate and allow for big pharma to do the longer term development. But you've also got companies like Karuna who are trying to do the whole bit, right? To do both the discovery as well as the clinical development and ultimately the commercialization. And I think that was, the approach Sage had. And I think we're no longer, they're no longer the exception. I think that, it's now become more of a norm. I think there are many more neuroscience focused companies. It's not just, biotech is no longer just focused in oncology or disproportionately in oncology. I think it's becoming a bit more democratic because I think they see that success is possible in the other disease areas.
Perfect. Sohini, you had your hand on.
Yeah, I was going to say that it's a really interesting discussion about the role of biotechnology and innovation in CNS. And speaking from the Parkinson's perspective, I think we clearly have seen that as well. The percentage of biotechs who are really spearheading sort of aggressive, innovative thinking and risk taking has absolutely increased. I do think, though, it warrants saying that in order to, CNS is just harder than oncology, I think, for vast reasons that we don't have to really go into. And I would just say that at least in our experience in Parkinson's, to be able to give these biotechs the greatest success, it actually helps to have a group like the Fox Foundation, and this doesn't need to be a nonprofit. It can be, this role can be played by others, whether it be government, et cetera. But the point being, somebody to help kind of provide access to resources, whether it be animal models, whether it be viral vectors or assays, whatever it may be, to be able to kind of put groups in contact with KOLs across different realms. We have a lot of experience working with biotechs who are working in the preclinical phase, but they're already thinking, what are my next steps? And they know the KOLs when it comes to kind of that particular stage of research, but they don't know who the clinical researchers are that they may want to bring on board to look at their program and advise on what are the phase, how to design the phase 1 studies, what is the right population, how should they think about a regulatory label, et cetera. And that's where it's really helpful to provide groups that have that ability to assist it, because at the end of the day, the wonderful thing about biotechs is that they're aggressive and they want to go forward precisely for the negative reason, which is that they don't have enormous resources. So they're focused on getting there as quickly as possible. But the resource issue also is an inhibitor. And I think where we've seen success for biotechs is when there is an entity that can help them bridge that sort of resource gap, whether it be knowledge, access to tools, funding, or a combination thereof.
Thank you, Sohini. I'm going back to the Q&A in the chat. So the next question comes from Jesus Giraldo from Universitat Autònoma de Barcelona in Spain. And his question is about how to solve the brain disorders, going back to the science, how we can connect the different research approaches at the receptor level, such as drug receptor interactions and receptor signaling pathways with brain circuits. So how to go from receptor activation to circuit activation, bridging the gap between the different scientific disciplines involved when researchers are so much specialized.
Yeah, it's a great question. I'll talk a little bit about some of the things we're doing at Karuna, because we have both a molecular pharmacology-based approach focused on the muscarinic receptors, but we also recognize that to complement that, we perhaps need polypharmacology approaches as well. And we're pursuing that through our collaboration with Psychogenics, because we recognize that the disorders may not represent only a dysfunction that can be corrected through the modulation of a single receptor, but in fact may require modulation of broader circuits or broader collections of molecular interactions, molecular consequences, right? I think many of the diseases that we are pursuing are complex in their etiology, and they don't simply reflect a single genetic lesion or a failure in a single receptor system. And so at least one approach that that we've tried to take on is using behavioral phenotypic data to guide the medicinal chemistry optimization, again, as a complement to molecular receptor-based single-targeted approaches, realizing that different drugs may be necessary for achieving, having an impact on these complex diseases.
Great. So we have a wonderful question online. With COVID, we saw a lot of misinformation with the social media and otherwise. So there is information and there is misinformation. And the question really is, what can we do for CNS diseases to make sure that we can engage the patients the right way for the right trials and not be hampered by misinformation, build the trust in the public and in the patients into the science that's being done in order to engage them to enroll into clinical trials. And maybe, Sohini, you could start with the Parkinson's Progressions Markers Initiative and what's being done to engage patients there, as well as the patient-based information that you are gathering and how we are giving the information back to patients to engage them and share the science with them.
Sure, I'm happy to start off. I think at the highest level, the answer to that question is open and transparent communication. I don't know how many people would find this shocking, but in this day and age, most people who engage in clinical trials actually still never receive the results of the trial they were in. They never receive any communication from the investigator, the site, the sponsor, any sort of entity involved about what the outcome is. It's the minority of individuals who receive any information. So that just gives you a little bit of a sense of where we're starting from in terms of like the ballpark sort of situation. And I think that when you expect patients to be partners, which frankly in clinical research they are, because at the end of the day, without their participation, there is no clinical research. They are probably the most essential partner. I think that you have to start off thinking of them as an actual valuable and valid partner. And that means that you are explaining why this particular approach is in clinical testing. What are the data that justify getting into the clinic? What are the benefits and the risks in a way that is easily accessible and isn't a 15-page, buried into a 15-page consent form, which is intimidating and doesn't really allow maybe for many individuals to understand really the nuances. To encourage them to talk about the study with their loved ones, with their primary doctor, to not place emphasis on an individual having to make a decision here or there, but it's their right to actually think about it and ask questions, etc. So, and then to continue to give progress over the course of the study. And by progress, I'm not saying, share the data, but explain, there's a delay. We haven't been able to recruit people. So we thought this would be over and we'd be able to share results, but we can't. We're still in the recruitment phase, or the data set is now locked. Analysis is high. But these kinds of high level things make an individual feel that you actually value them and that they are essential and that they're a partner to your efforts. And I think, there have been efforts made for this concept of patient centricity in research. I think is still very much a buzzword, but within some groups, it is becoming more of a reality. And I think you mentioned the Parkinson's Progression Markers Initiative, or PPMI for short, which is an observational study that is sponsored by the Fox Foundation. And I think it provides a really good real-world example of how treating an individual as a participant actually benefits the study. Now, in this case, it's an observational study. You would actually argue it's far harder to get a person to stay involved in an observational study, particularly since it's now been going on for 11 years and there's no drug being tested. And we're asking an individual to undergo like 7 to 10 lumbar punctures over the course of their participation, in addition to multiple imaging and multiple other biosampling, et cetera. But we have about 85% retention rate across the study, and it's a global study. And if you ask why, it's because from the beginning, we treat them as partners. We go into huge detail about what the point of this study is. We have things called retention events that are held at sites every year, where the site investigator and the Fox Foundation representative go. And we actually talk about where the study is. Participants get a packet, IRB approved, of all the abstracts and papers that have come out of the data that they have contributed. They're able to see the usage stats, in this case, over 8 million data downloads since the start of the study. They're able to actually see what's coming out of it, and it gives them more of an incentive to stay in it because they actually see the output. They feel a great deal of pride in being able to contribute to research. And, we're able to kind of tell them, honestly, there's over 20 Parkinson's trials in phase 2, either leading into phase 2 or have been in phase 2 or currently in phase 2, that have actually modeled themselves on data coming out of this study. And I think that's the greatest satisfaction they have. So it's a great real world example of where just really honestly, simple, open, transparent communication and recognizing a clinical trial participant as a true partner, it's a game changer, and I hope we're seeing more fields and more stakeholders really take that up.
Thanks, Sohini. That was great. This was a question from Sherry Butts, I should have mentioned. Chas, you had your hand on.
Yeah, Chas Kalpana, this is the last part of the discussion because incredible, but we finished our time. It was very fast. Sorry.
Do you want me to respond?
Yes, go on.
Yes, please.
I think all I was going to say is, Sohini is obviously a brilliant communicator, but we do need more trusted storytellers. We need scientists who give patients hope, who are optimistic. We are in an era where, brain diseases are going to be transformed in the next few decades because we've got these new technologies and new modalities, et cetera, et cetera. Kalpner at the start talked about the amazing successes, in SMA, DMD, migraines, schizophrenia, Alzheimer's. We need people telling these stories. I think the other thing we need to do as a scientific community is absolutely always support our organizations, scientific, FDA, NIH, United Nations, et cetera, et cetera. We need to do everything we can to support them so that collectively we're giving out a more uniform message. We're always going to have the challenge of diverse views and social media, but we need to respond to it collectively. So thank you.
Thanks, Chas. Should we give the final 5 seconds to Julia Mabel, if we have time?
Yes, very fast, because we have to end and explain in the wrap up the next networking session, and we have a few minutes. But go on. It's very interesting.
Thank you.
Go on, and Julia, you are the last for today.
Thank you. I'll be very short. Yeah, one of the things is actually it follows both on Sohini and on Chas, which is we just need to talk about the science that we do to people. It means engaging. It means that if a journalist calls us, even though we are busy because we have 1,000 grants to apply for and papers to submit, take time and try to be clear and, try to educate ourselves in how to talk to people about science. Talk to your sons, daughters, family members, kind of like get that ball rolling, because that's really important. Because unfortunately, one of the flip side of open science is that it can be misconstrued. And we know what's happening with various conspiracy theories, et cetera, that claim to be based on science. So we can't be illuded that we are better than that and that we're separate from that. So just talk to people about science all the time.
So thank you very much. Thank you all of you. Really, I feel really privileged to have the opportunity to moderate and to listen. Although so far, but I feel all of you very close together and also thinking very nicely. So unfortunately, we have to finish because it's time for us to wrap up now. And it's really difficult to wrap up so many great ideas coming from incredible leaders like you are. But if I could say just 2 points, that it seems that although, of course, I agree with Sohini that it's a little bit difficult and naive to think that is the same, and you can go from COVID-19 to these brain diseases with a plethora of diseases. But it's clear that some conclusions change, because I was trying to write conclusions from our table, right? So some conclusions change, and really, all of you are combining a lot of ideas, focus, leadership, global prioritization across the field, just not into the boss, in our boss, combining open and no open science. It's not clear idea, but it's in our this discussion, right? Including all stakeholders. I was thinking that it's like these open science networks that just always says, also in Santa Fe, right? That now it's clear that is the way that could generate. I don't know, but something I could was listen very interesting words, your very interesting words, and I was thinking that it's like micro factories focused in something in patients, some population, some targets, but microstructures, flexible, but very inclusive, with stakeholders from patient-centered idea to quick validate or not validate and go on to the next. I don't know, in a new way, as Sohini says, to go faster and to stop before to know for many years after that it doesn't work, to have some kind of companion stratification in parallel with the drug in the target or the mechanism, right? So, but it's difficult, and it needs to write very conscious and to read by all. But I have a good news about this, and is that Julia Meliak offered the editorial of his journal to publish this, and I would like to ask all of you, the global community that are today, to try to create this kind of global effort for brain diseases and try to be connected. The next connection is this editorial, but maybe to generate with all the stakeholders new models to collaborate in this democratizing science that Jim mentioned. So we have to stop, but we will continue thanks to Julia Meliak. Thanks. I don't know, Julia, if you want to say some words about this.
Yes, of course. So the plan for us will be to publish a summary of this discussion, but also to make it bigger. So for it to be kind of a call to arms for everyone who would like to participate. Obviously, for the company of biologists itself, our involvement will have to end there because we're a publisher. So we will publish this article. We will make it really available to everyone interested And then we will help with promotion. But then hopefully the scientific community will rally behind these very positive and very encouraging thoughts, so that there will be a renewed sense of urgency to bring all of the brilliant minds together and actually kind of break the silos and make tangible progress.
Exactly, in this kind of open science. Thank you very much. So we are finishing. Thanks to all. Really, it's a privilege to have all of you here. And now I would like to invite you to join us for further discussion now in this remote networking lunch. I'm reading the text that was given to me by the Keystone Symposia. That is that you have to follow the link on the event homepage to connect with colleagues around the world in a small group discussions to strike up new collaborations. So you will be receiving an email from Keystone Symposia as we speak. Probably we already have it with the link and instructions to join. So check your inbox for detailed instructions. So our idea was this idea of Remo, but for me it's new also. It's like this cocktail after event that we usually use to talk together and with the speakers that go into and out of the tables. So thank you so much again for joining us in this virtual Keystone Symposium ePanel event. A recording will be available shortly in the event homepage. Thank you very much again. Love for all of you.